Today I'm going to discuss the lactose operon and inducible operon. But before that, let us first define several terms regarding this discussion. Operon. An operon is a group function-related structural genes that allows the synthesis of protein to be controlled coordinately in response to the cell's needs. Two types of operand: inducible operand and repressible operand. Inducible operand is an operand wherein the effector molecules activate transcription. In this system, lactose acts as an effector molecule. Lactose is a disaccharide containing one glucose and one galactose residue. Moreover, the topic I'm going to discuss is an inducible operand, which is the lactose operand. On the other hand, a repressible operand is an operand wherein the effector molecules repress transcription. An effector molecule is a molecule that interacts with the repressor and affects the affinity of the repressor for the operator. Now, let us get to know what the lac operand is. Lac operand, or the lactose operand. It contains genes encoding for proteins required to metabolize the sugar lactose. It is found in the E. coli or Escherichia coli bacteria, which is a prokaryote. There are two scenarios of lactose operand. Absence of lactose. If lactose is absent, the repressor is active and no transcription will occur. Presence of lactose. If lactose is present, the repressor is inactive and transcription occurs. The lac operand, absence of lactose. The lac operand consists of various components. LAC-I is a regulatory gene. The regulatory gene codes for regulatory protein. The lac repressor, encoded by the LAC-I gene, is a regulatory protein of the lac operand. mRNA or messenger RNA codes for beta-galactosidase, lacrimase, and transacetylase. LAC-B is a promoter gene. It is where the RNA polymerase specifically binds during the initiation of transcription. RNA polymerase is a single unit protein that acts as a carrier for the entrance of lactose into the cell and transcribes all bacterial operands. LAC-O is an operator gene. The operator is located in between the promoter and structural genes. It serves as the binding site of the repressor to terminate transcription. LAC-Z, LAC-Y, and LAC-A are the structural genes. The operand encodes one or more genes for inducible enzymes. The lac operand encodes enzymes necessary for lactose metabolism, including LAC-Z codes for beta-galactosidase, LAC-Y codes for beta-galactoside permease, and LAC-A codes for beta-galactoside transacetylase. Beta-galactosidase degrades lactose into galactose and glucose for the production of energy, while some are converted into allolactose. Beta-galactoside permease functions in the transport of beta-galactosides from the environment and into the bacterial cells. Beta-galactoside transacetylase is also involved in beta-galactoside metabolism. It is an enzyme that also needed to break down many sugars related to lactose. Without lactose in the cell, the repressor protein is active and binds to the operator. It prevents the read or transcription through RNA polymerase into the three structural genes, so no transcription is possible. In this case, the lac operand is off. The lac operand, presence of lactose. With lactose in the cell, lactose or allolactose binds to the repressor, which makes it inactive. Allolactose acts as an inducer by binding to the repressor, resulting in a conformational change in the repressor and losing its affinity for the operator. It closes off the operator, clearing the way for RNA polymerase. Thus, RNA polymerase can then bind to the promoter and transcribe the structural genes. Transcription of structural genes occurs at a higher rate, resulting in an increased concentration of products. In this case, the lock operand is on. Eventually, if all lactose is used up, there will be no more allolactose and the operand has turned itself off. The lock repressor reverts to its original form and can now bind to the operator, so mRNA transcription stops. That ends my discussion. That's all, and thank you.
Hey there, for this video we will be discussing about the tryptophan operon. But before we delve into the discussion proper, let us first define the terms that will be used in the discussion. So first, of course, we have the term operon. So what is an operon? An operon is a group of functionally related genes that allows the synthesis of protein to be controlled coordinately in response to the cell's needs. There are two types of operon, an inducible operon and a repressible operon. So an inducible operon is an operon wherein the effector molecules activate transcription. So the effector molecules helps in the transcription process to occur. Whereas the repressible operon, the effector molecules does not help. Instead, it helps in repressing the transcription process. So the tryptophan operon, as used in this discussion, is a type of repressible operon. So in the next few moments, we will know why the tryptophan operon is a repressible operon. Okay, the TRP operon, or the tryptophan operon, abridged as TRP, is a group of genes that encode biosynthetic enzymes for the amino acid tryptophan. So, saying the tryptophan operon doesn't directly produce the amino acid tryptophan. Instead, it encodes biosynthetic enzymes so that the amino acid tryptophan will be produced. So the model or the tryptophan operon that will be discussed in this video is the tryptophan operon found in the E. coli bacteria. So it is a prokaryotic tryptophan operon. Here we have a simplified model of the tryptophan operon. So as you can see, the tryptophan operon is not just a single point in a DNA. Instead, it is a whole set of structurally related genes. So they serve one purpose, and for this, they serve a purpose to encode the amino acid tryptophan. Here we have a red colored box. So this is the regulator gene, or the TRPR. So the regulator gene is the site wherein the repressor is encoded. Also, we have the promoter. So the promoter, as we all know, is the binding site for the RNA polymerase, which is the primary enzyme for the synthesis of the mRNA, or the transcription process. Next, in between, or within the location of the promoter, we have the operator. So this is the binding site for the repressor. So the repressor that will be produced, that will be encoded by the regulator gene, will bind here in the operator. So it's the TRPO. And then we have here the TRPE, D, C, B, and J. So these are the structural genes. So these genes will code for the biosynthetic enzymes that will help in encoding the amino acid tryptophan. So TRPE and D will code for antranilite synthetase. TRPC will code for glycerol phosphate synthetase. and trip B and trip A will code for tryptophan synthetase. So with these enzymes all together, the amino acid tryptophan will be encoded. In the tryptophan operon, the transcription process has two scenarios. So the first scenario is wherein there is a high tryptophan presence in the cell. So when there is high tryptophan, the repressor is active. So when the repressor is active, of course, the transcription process will be repressed. Therefore, there will be no transcription. However, on the other hand, when there is low tryptophan in the cell, the repressor is inactive. Therefore, transcription occurs because there is no obstruction in the synthesis, in the transcription process, or in the synthesis of the biosynthetic enzymes that will code for the tryptophan amino acid. Okay, here we have the first scenario wherein there is a high tryptophan in the cell. So as you can see here, we have these molecules over here. So let us explain. So this one is the repressor. So it is encoded by this gene, which is TRPR, the regulator gene. So the repressor is powerless in itself. So even if it binds here in the operator, the RNA polymerase will not recognize the repressor as an obstruction in the transcription process. So what will happen is it needs something, the repressor needs something to activate its function, that is to repress the transcription process. So as we can remember from the previous definition of the two types of operon, we said that the tryptophan operon is a repressible operon wherein the effector molecule helps in repressing the transcription. So our effector molecule here is the tryptophan. So the tryptophan is the co-repressor. So it helps in repressing the transcription process. So the tryptophan, for the repressor to be active, it binds to the repressor, and then the repressor now will be active. So the RNA polymerase comes in. Okay, so the repressor now is now active. So the RNA polymerase comes in and it sees an obstruction. Therefore, transcription doesn't occur. So the reason why the tryptophan is a co-repressor when there is high tryptophan in the cell environment is it regulates it regulates in keeping the concentration of the tryptophan molecules in the cell at a regulatory level. So when there is high tryptophan, it sees that it doesn't need too much, so it stops the production for surplus to not happen and all. Next, we have the next scenario, which is low tryptophan. So here we cannot see any tryptophan molecules in the environment. Therefore, the tryptophan repressor is inactive. So it doesn't matter if it binds to the operator or not, because it is still inactive. So the RNA polymerase binds to the promoter, and then transcription occurs. That is all. Thank you.